Here we want to look at how the affinity of hemoglobin for oxygen can change relative to the environment of the tissues which it passes through. At the top here, we have our carbonic acid equation. And what it shows is that tissue carbon dioxide production, a byproduct of aerobic respiration through the Krebs cycle, as it increases, it combines with water in a reaction catalyzed by the enzyme carbonic anhydrase inside red blood cells. This produces carbonic acid, which rapidly dissociates into a free bicarbonate ion and a free proton. And so our metabolizing tissues are always doing this. They're always producing carbon dioxide. They're always producing some free protons. And that generates the pH that's normally in this tissue. Now, when metabolism rises, we get an increase in CO2 production. This causes a rightward shift of this carbonic acid equation. It's called the law of mass action. This will generate an increase in the number of free protons, which is related to a decrease in pH, or an acidification of that tissue. We could also be producing lactic acid during an increased metabolism. That also would acidify this tissue. If we come down to the bottom here, again, we're going to look at the percent saturation of hemoglobin on our y-axis and the partial pressure of oxygen on our x-axis. At the lungs, we're at a partial pressure of oxygen in the alveolar air of about 100. Well, that generates our near saturation of hemoglobin with oxygen. We then send that blood off to the tissues. If those tissues are at rest, they're generating a normal number of free protons, and we end up with a physiological pH of about 7.4. This results in a certain amount of unloading of oxygen at that tissue, as we've seen previously. However, if we increase metabolism, we get more CO2 production, we get more free protons, and those two molecules bind to hemoglobin as allosteric modulators, and they affect the affinity for oxygen, causing it to release even more. And so what we end up on is a rightward shifted curve here at a lower pH. And so if we pick a tissue PO2 of about 30, we go upwards from that, what we would find is that on the red curve, right, we would have a degree of saturation or desaturation down to about 65 or so percent. Whereas on the right shifted acidified curve, we're down here at about 50% saturated. We have enhanced unloading through that allosteric modulation of the free protons and the carbon dioxide binding to hemoglobin. Well, it can get even better than that. We also, in many tissues, have an increase in temperature when their metabolic rate goes up. This is especially true in skeletal muscle. The production and use of ATP wastes energy that is lost as heat. If we can't remove that heat as fast as it's generated, the tissue will actually warm up. Again, skeletal muscle is known to do this. And so therefore, that rise in temperature, that rise in thermal energy, loosens the connection between oxygen and hemoglobin, causing a greater degree of unloading. Finally, we have another allosteric modulator. This is produced as a byproduct of glycolysis, this BPG molecule. Well, glycolysis is always running, right, generating this molecule to some degree. When metabolism goes up, the production of BPG rises. It's stimulated by hypoxia when glycolysis would be even further enhanced. And we end up with more of this negative allosteric modulator, causing an increased rightward shift. And again, we could draw a right shifted curve here, where they would have even lower percent saturation, enhancing unloading. And again, we're adaptively matching the metabolic activity of the tissue to the oxygen delivery through microenvironmental changes caused by that rise in metabolism.